Hello, everyone, and welcome again to an Ageless Partners information session here. We're here with Kamila, and she's going to be speaking about multi-omics technologies. Kamila has a background in nutrition and bioinformatics and biology, and she's very, very interested and knowledgeable about this subject. So we really are looking to figure out, what is multi-omics and what can we derive as actionable insights to help ourselves and our audience live longer and enhance and slow down their aging. So thanks, Kamila, and I will let you take it away.
Thank you, Jason. I appreciate the opportunity to talk about this topic, and thank you for joining me today to discuss multi-omics technologies in aging longevity research. If you have any questions, I'll be happy to answer them at the end of my presentation. So without further ado, let's just start by my outline. Today, I'd like to identify what is omics. I'd like to identify why do we need it, what biomolecules are used by it, how any individual can benefit from those, and some of the shortcomings are also going to be discussed in this presentation. This picture illustrates the hallmarks of aging. And what we can see in here is that there are a lot of processes such as dysregulated nutrient sensing, altered intercellular communication, epigenetic modifications, decreased stemness of the cells, loss of proteostasis happening at the same time in our organism. So that means that aging is really a multifactorial process. And we see that senescent cells are accumulating in our body, and a lot of DNA damage markers and epigenetic modifications happen to our DNA and telomere length shortening. So we need to be able to analyze all of those processes simultaneously. That's why we need the multi-omics technologies. And aging is also, so again, aging is multifactorial and continuous process. It is also heterogeneous among people because due to the significant environmental effect on our DNA and our aging, the way we age, it is heterogeneous among people, and organism also ages differently depending on the type of tissue. And that's why new tool is necessary to account for all those factors. And generally, what is omics is a collective name of branches in biology that research genome, epigenome, transcriptome, metabolome, and there are new emerging omes appearing nowadays, but those are not the scope of my presentation today. Overall, in aging and longevity research, there are several types of biomarkers that are informative. For example, again, talking about that picture, like before, that picture was depicting molecular and cellular biomarkers happening that are informative for aging and longevity research. And mostly they are used in assessment of cell functionality, whereas and they are informative molecular and cellular biomarkers, informative in early stage cellular damage events, and also in accumulation of the deleterious changes in cells and tissues, whereas systemic biomarkers are mostly informative, mostly used in clinical settings, and they are mostly informative for late-stage pathological processes to reveal what types of conditions and what diseases are people suffering from. And also physical and functional and cognitive performance testing is also informative in late-stage pathological processes and diseases. What's good about omics-based biomarkers is that they are actually continuously informative. So that means they are informative as at early stage and in late stage in processes and diseases as well. So overall, if we will use all of those, if we will have the capacity to use all of those information, and we will be able to eventually reveal the aging profiles and so-called ageotypes of people, because again, people age differently, and that's what omics-based biomarker is really useful for. So again, omics-based biomarkers can be equally applicable throughout the human lifespan, and they are useful from single cell profiling to systemic phenotyping, and they can be divided, as we see, as genomic, epigenomic, transcriptomic, metabolomic, and some other omics. What types of molecules are used in multi-omics research? So here we see the types of molecules like DNA, RNA, protein, and metabolites that are used mainly in single omics, which is a branch of omics that are usually concerned with a particular type of tissue. But what's good in multi-omics is that we can just analyze the set of those molecules. So for example, when we talk about DNA in multi-omics research, it's going to be genome, similarly transcriptome, epigenome, proteome, and metabolome. So usually nucleotide polymorphisms and copy number variants and genomic rearrangements are used in multi-omics research. Those are generally the processes that make our genome unique. When we talk about RNA, it's usually researching gene expression and alternative splicing on non-coding RNA that are the processes, alternative splicing and non-coding RNAs are the things that regulate our gene expression. There are some other modifications that are happening to our DNA or even to protein, for example, histone modification, also chromatin accessibility, transcription factor binding, and microRNAs that are also influencing to the level of our gene expression. And finally, protein and metabolites are usually researching for protein expression and modifications that are happening to the configuration of the protein, for example, or cytokine array, which basically is the marker of the inflammation happening in an organism. And when we talk about metabolites, it's usually the products of our microorganisms in our organism, and usually we search in serum, urine, plasma, profiling, and etc. Genome. Our genome, human genome, consists of like 20,000, approximately 25,000 genes, coding genes, protein coding genes, and genetic markers reflect predisposition to certain aging phenotypes. Although aging is only approximately 20% heritable, and now there are studies that suggest that it is the genetic contribution is even less, to approximately 16%, but some age-related disorders are genetically predisposed. For example, Alzheimer's disease has a heredity of more than 70%. Osteoarthritis and cataract have a heritability of 50% prognosis. So that's why research in genome is really important, because there is an overlap between disease and between aging and age-related diseases, and that's why it's really informative to understand the processes. There are three types reported of longevity genes that we really need to be aware of. So, for example, sirtuins, they are NAD-dependent enzymes. They are responsible for stability, activity, and localization of proteins. And they are also, they are usually responsible for removing acyl groups from our proteins. So that means that they really influence on the configuration of a protein. And there is a particular sirtuin 6 that are responsible for DNA repair. So that means that it is crucial for decreasing the effect from DNA damage happening in our organism. There is also AMP-activated protein kinase, which is responsible for the energy metabolism in our organism. It increases fatty acid oxidation and glucose transport. It releases additional energy from available or stored resources. And mTOR, you probably all heard about mTOR, which is a mechanism that regulates growth, and it signals to the body that they should grow when the time of plenty and not grow when food is scarce. So what we can derive from it, there are mechanisms that stimulate or inhibit mTOR, and you probably heard about, for example, mTOR stimulation ways as calorie restriction, and it also activates AMPk, which we've been talking about before. We can also, you probably heard about rapamycin or berberine and resveratrol, which are really popular compounds that mimic calorie restriction, and they are also mTOR. Actually, rapamycin, is an inhibitor of mTOR, whereas berberine and resveratrol, they act as mTOR stimulants. What we can derive from what stimulates AMPk and sirtuins. Stimulation can occur with the help of omega-3 fatty acids in diet, and of course exercise and calorie restriction. Whereas inhibition, AMPk stimulation, occurs again with calorie restriction and exercise, intermittent fasting, and also cold exposure, because it really slows our metabolism and lowers inflammation. What actionable insight can we derive from the genome? We should avoid DNA damage. That means that we need to use sunscreen and avoid X-ray, and especially in winter, for example, when sun is really hazardous. We also need to keep a limit in our calories and restrict the leucine-rich products. Consume enough polyphenols and omega-3 fatty acids, and we need to also regularly exercise and keep our metabolism slowed down a little bit by, for example, keeping cold. Epigenome. DNA methylation is the most common epigenetic process, and it's often and it usually influences by lowering the gene expression. Other epigenetic modifications, such as histone methylation and acetylation, have been linked in addition to as a modification in addition to DNA methylation. There are also environmental and lifestyle variables that influence our genome. For example, here I'd like to speak how the epigenetic modification happen. So here we can see the chromosome, and we can see that histones, that are proteins, are actually wrapped around the chromosome. And what happens during epigenetic modifications is that epigenetic factor is gets attached to histone tail, and it can be a methyl group or acetyl group, or sometimes even glycans, which are like carbohydrates, and it actually influences on the accessibility of chromatin to the gene expression. That means that when our proteins are too occupied by epigenetics factors, that eventually influences to the lowering of gene expression. So connection between lifestyle and epigenome. Again, several lifestyle factors have been identified that might modify epigenetic patterns. So you've probably heard a lot about diet, obesity, physical activity, tobacco smoking, alcohol consumption, environmental pollutants, psychological stress, and working on night shifts. Those are critical factors that are influencing our epigenome, and we need to consider those because environmental factors approximately have a 70% influence in our gene expression. Transcriptome. So as we know, genes are transcribed into RNA molecules for DNA replication or translation. Transcriptome is a collection of all RNA transcripts. It is separated between coding RNAs and non-coding RNAs, which are usually responsible for regulation of gene expression. Here we can see the typical result, for example, of an analysis of proteome, and it was taken from a Swiss Institute of Bioinformatics. We can see the blue upregulated protein expression, and in red we can see proteins that are downregulated in aging. And mostly what can be seen is that processes that are responsible for cell adhesion and interactions, metabolism and signaling pathways, regulation at the organism level, meaning homeostasis, development and growth of organs and tissues, again, mTOR, immune system and development and growth of the body are compromised when aging happens, according to the KEK database. Metabolome or microbiome. Metabolome is the microbiome is a full set of microbial species harbored by the human body. And the gut has the biggest microbial population, with microbial cells and genes outnumbering human cells and genes. Although twin studies have discovered a little genetic effect on some phyla, the majority of variance is due to environmental factors. So actually our human, when we compare human cells to microbial cells, there is an evidence that microbial cells in our body are 10 times more than our human cells. And that's why bacterial genomic contribution is critical for human survival. And human microbiome actually contributes approximately to 8 million unique protein coding genes. And if we remember how much protein coding genes contributed by our own DNA is 25,000, when we compare it to the contribution of human bacterial contribution to the protein coding genes, it's 360 times more than contribution of human genes. That's why actually metabolome and microbiome is a really significant factor contributing to our genome. So what we see that researchers found significant changes in several metabolic pathways, and mainly in vitamin E metabolism, tryptophan metabolism, acetyl coenzyme catabolism, and namely vitamin B5 synthesis, lysine metabolism, and aspartate and asparagine metabolism, which are generally, for example, tryptophan is responsible for the protein synthesis, and acetyl coenzyme is for the energy. Metabolome or microorganisms are important for our immunity. We can see that in metabolism and neuronal development. Again, microbes are responsible for synthesis of crucial vitamins, for example, group B and K. Diet alters the composition of a microbiome, and insufficient fiber in diet leads to an increased syndrome of leaky gut, and which eventually might lead to the inflammation. How to make a little bit off top. So there are a lot of studies that are trying to see the processes how to make a mouse live longer, and usually they suggest that we need to reduce the amount of time the mouse eats. It means that we need to fast, we need to restrict amino acids intake, and we need to slow down the metabolism by keeping our body cold. We can also influence on the composition of our microbiome. So there is a study that actually showed that intermittent fasting really influences on the dynamics of our microbiome during the day. So for example, here we can see that there is a normal mouse which has been eating a lot of fiber, and we can see that population of the gut bacteria is really dynamic during the day, and that leads to the lean mouse. So this mouse has no obese conditions suffering from. And on the other hand, we can see that there is a same mouse that whose diet is insufficient in fiber, and we can see that the obesity-associated gut bacteria population exists in the gut of this mouse. And when we, for example, do the time-restricted calorie intake, even if we consume not insufficient fiber, there is a dynamic change in the gut bacteria and reduced obesity effect on the mouse itself. So that means that there is a really positive effect of intermittent fasting on our gut population. But there are also implications. We need to consult a doctor before doing so, of course. And probably there is a positive effect due to mTOR regulation, because mTOR regulates how much protein we consume. And when we do the restricted calorie intake, it decreases mTOR signaling, and that's why it may be contributing to the slowdown of metabolism eventually. And there are also compounds that are useful in calorie restriction, like diet mimicking, for example, rapamycin. Now I'd like to talk about Omics Geography and Market trends. We see that there are a lot of companies nowadays that are involved in researching omics-based biomarkers, and mainly they are located in US, 60% of them reportedly. There are approximately 12% located in Europe, and there are some companies that are located in Singapore, Korea, China, and Australia. When we see the distribution of those companies, we can see that there is a popular trend among companies to develop research kit and genetic tests. They are becoming really popular nowadays, and with the decrease of the sequencing cost nowadays, anyone can order at-home test and do the genetic test at home. There is also an increasing trend in software development and data management or data manipulation apps among omics-based biomarkers. But of course, I think that this trend will be changing, and there will be more companies that are developing software for the healthcare in the next few years. For example, this data is for 2021. There are less companies that are developing pharmaceuticals and devices, but there are a lot of initiatives nowadays to do wearables. And finally, we will be able to see probably the completely different picture in next few years. Here we can see the distribution of those companies again. We can see that mostly they are directed to the diagnostics, to the research and development, therapeutics, and digitalization. And we can see that a lot of companies are targeted to the diagnostics of some age-related diseases. However, there is also a huge number of companies that are digitalizing their the aging research, and there is a an emerging trend in digital biomarkers that are happening in next few years. And here is another pie chart that illustrates that 50% of companies are involved in biotech, 17% of companies are developing at-home tech, and 17% are involved in service development and clinical tests. So there are a lot of, despite the advantages brought by omics technologies, there are shortcomings. So for example, they are still costly and require specialized equipment and also highly trained employees to operate. There is also limited availability of multi-omics data sets for the same samples. There is also an inconsistency. For example, genome and transcriptome can be measured with a complete collection of variations, whereas there is a limited data for proteome and metabolome of human. And there is a complexity of the organism that should be considered. For example, genome remains relatively constant between cell types. However, other types of omes, for example, metabolome, are dynamic throughout the day. And also another shortcoming is that, as we can see from this slide, all of the omes are interconnected between each other. And that's why it's really hard to analyze the data independently. And for example, it can skew the results. And there are a lot of researchers that are, for example, discovered certain amount of metabolites that are linked to age, but when we try to independently research those linked metabolites, it happens that less number of those are exactly linked to the aging. So that brings to the end of my presentation, and we see that omics-based tools have a lot of advantages, and eventually when we solve the shortcomings of omics tools, we will be able to probably more understand the molecular mechanisms behind the aging, and I hope to see that in near future. And thank you for your attention, and I'm sorry for being probably confusing sometimes, and I'll be happy to answer your questions if you have like any. And thank you so much. Oh yeah, and by the way, what advices can we get from this presentation eventually? We should again avoid DNA damage. We need to restrict our calorie intake. We need to restrict leucine-rich product in our diet. We need to consume enough polyphenols, meaning that we need to eat more berries, more grapes, and omega-3 fatty acids that are usually consumed from fish, for example. Regular physical activity. We need to keep cold, eat more greens, avoid tobacco smoking, avoid alcohol consumption, reduce environmental pollutants because it really influences on our toxic on our liver. Reduce psychological stress, working on night shifts, and of course we need to monitor our health and maybe even try to make a genetic predisposition test to see our maybe intolerances, if we have any intolerances or genetic predispositions that we need to really monitor and be aware of which. So thank you for your attention. I'd be happy to answer your questions.
Thanks, Kamila. Really fantastic. Well, I had two quick questions.
Sure.
The first one, if you could go back to that slide that had the rats on it, and it was talking about that one right there. So are the green dots quality bacteria and the red are?
Yes. So these are usually gut bacteria, in green, there are the bacteria that are usually responsible for the permeability of the membrane. And green and blue population are bacteria that are beneficial for us. Whereas the red bacteria, we can see that these bacteria are like obesity associated, and that's why we need to like dynamically shift the bacteria in order to during the day be able to have the positive effects and negative effects. I mean, like positive effects of the beneficial for us bacteria. So yeah,
Gotcha. Interesting. And the picture on the bottom there, where you have red dots, dark and red light, just comparing the two bottom pictures, I mean, does that, like dark red, does that infer that those are worse than the light red, or?
Oh, no. So light red are obesogenic bacteria. Obesity, I mean, they are like both obesity associated, but some light red are obesity like enhancing, whereas dark red are like good in terms of that they so it's written obesity protective bacteria.
Gotcha. That makes sense. So those are good, but not as good as the green and blue ones, though, likely, right?
Yes.
Okay, that makes sense. I mean, given that they're restricting their window of eating, but eating the same thing here, you get a little bit of a benefit. Nice. Okay, that makes sense. One other quick question. If you go to the slide that had the vitamin K, vitamin B, I wanted to take one quick look at that.
This one or this one?
Right here, yeah. So it says microbes are responsible for synthesis of vitamin B and vitamin K. So
yes.
vitamin, just a quick question. Vitamin B would be a water soluble, right? And vitamin K would be fat soluble. So I don't know, is there any like relation there? I would think it would be like, how would you kind of differentiate between those in this example here?
Oh, in this example, well, here I included vitamin B and K because like bacteria, when they ferment like our food we eat, the metabolites of those bacteria are vitamin B and vitamin K. And yeah, they are different in terms of that. Yeah, B is water soluble, but it depends. The difference is that the way we use those like vitamins. So for example, if we have an excess of vitamin B, water solubility will help us to like get rid of the unnecessary vitamin B parts through the like urine, and whereas vitamin K will help us to get it through the liver. We need to like make it water soluble in the liver and then again through the urine.
Interesting.
So in that sense, there is a difference, but yeah, I don't know about the difference between like the microbes.
Right. No, that makes sense. I think that's a good comparison. And so it almost seems like the microbes would be consuming prebiotics, and then they would then, poop out postbiotics. And when
Yes,
they're actually consuming, maybe, they're synthesizing vitamins based on that.
yes.
Okay. So they're kind of utilizing those vitamins rather than necessarily creating them, or?
No, I mean that the food we eat, microbes use to like eat themselves. But when they are like excrementing, the product is actually the vitamin.
Interesting.
So that means that they really like produce the vitamin B.
That's interesting. So when I take a vitamin B supplement, the microbe is consuming that and then creating the vitamin B.
I'm not sure if vitamin B gets like consumed by the bacteria itself. Maybe it will like go as a ready product.
I see.
So,
Okay.
yeah.
So there could be some, there could be some interrelation there. Okay, that makes sense.
But that's a really interesting question, actually. I've never like thought about that. What happens when we like ingest the ready product?
Yeah, I mean, I've heard theories and they're just theories, but that our bodies can create certain vitamins. Now, vitamin B being water soluble, I feel like it could be kind of hard. Like I feel like you really need to get that from your diet. vitamin K, if it's fat soluble, it's stored in your fat cells. So if you're, if you're doing a fast, then you would then be burning fat and then having the microbes synthesizing that vitamin K to then
Mhm.
benefit your cells, essentially.
Mhm.
I would understand.
Yes. Yes, I think so.
It's complex. Awesome. Well, thanks a
Yeah,
lot, Kamila. And Trace, you got a quick question there?
yeah, I just want to say, first of all, really great job on the presentation, Camilla. Like you
Thank
mentioned,
you. Thank you
you're
so much.
welcome. You mentioned a lot of actionable insights, and that was really like a great build on the last presentation you did. I really enjoyed that. That's beneficial
Thank
for
you.
any viewers. So I really love that. And in terms of what you just mentioned, in terms of the fats and all that, it's really interesting because I don't know how the body will actually prioritize in terms of what fats it will actually excrete during fat burning, because when you actually ingest some fats, they can actually end up being stored for up to like 7 or 10 years, which
Mhm.
is why in inflammatory fats like omega-6 or oxidized fats can be a much bigger deal than just consuming a lot of carbs and sugar, because they can kind of be burned off quickly. But if you just have a lot of inflammatory fats sitting there for plenty of years, they would just contribute to just constant inflammation every single day for years and years and years. So
Mhm.
it's really interesting to think about how the body actually prioritizes fats, and if there's a way to actually make sure that your body actually prioritizes the most harmful fats first, instead of just going for whatever it pleases, or how it happens. Because like I haven't heard of any insights or any research on actually kind of focusing on specific fats of actually excreting those. Like
Mhm.
I know that glycine can actually be quite powerful to get rid of some fats from your organs, but still, like, there can still be a lot, and there can still be a lot of different parts that have different inflammatory profiles. So this whole aspect of focusing on fat burning is like, it seems like something that we don't have a lot of understanding about, but given the fact that they can just be stored for so long, it can be really beneficial to actually work all this out.
Mhm. Sure. Yes, actually, I need to mention that taking energy, like burning fats and taking energy from fats is actually really hard, because we need to like put our organism in a like keto to get the energy from fats, because usually it's much easier for our organism to get energy from carbohydrates we consume and from our muscles. And in that sense, in order to like get the energy from fats, we need to firstly put our organism in keto condition, like the keto diet, the most popular like ketogenesis. Like we need to put our organism into that, and maybe somehow we can. I don't know in terms of how to like differentiate between the fats, the types of fats we need to burn, because it's really hard to do that, if not impossible, I guess. Uh, but about glycine is really good for the permeability of our gut membrane, because it helps to thicken the wall of our gut. In that sense, it reduces inflammation. And maybe that's why it's good in terms of getting rid of overall inflammation in the body. And that's why probably it's really popular.
Yeah, it's a really good point. And glycine is actually like, I feel like it's actually one of the most important supplements to actually take, because other than actually being really good for your whole microbiome and gut health, it can actually, like, there actually been some studies on the fact that the effects from increased intake from like leucine and methionine, which really speeds up mTOR, which can be really detrimental, as you mentioned, if you just do it too much. And so actually increased glycine intake actually seems to be able to kind of offset this whole increased mTOR stimulation, and thus this effect can just mean that you can actually be able to kind of consume bigger amounts of proteins without speeding up your aging processes too much. And this is really interesting because today a lot of people they consume a lot of products that are really low in glycine, so they're just speeding up mTOR all the time, and they're just aging faster given that fact. Because earlier, many hundreds of years ago, we used to consume a lot of organ meats, which actually which actually contain a lot of glycine, and thus we didn't speed up mTOR that much, and didn't age as fast as we do today, because we just constantly just eating muscle meat that is just so low in glycine, and also like things like cheese and fish that just contain a lot of methionine and leucine, just speeding up mTOR all the time. So to me, like in our modern age, like consuming glycine, like either through organ meats or through a supplement, like it's really beneficial if you want to consume some protein without it having this big detrimental effect on just growing too much and just speeding up your aging too much. It really seems to have so much power there.
Yeah, that's a good point.
Yeah. So you're basically saying that you're able to get some of the benefits without eating meat, where you would normally have to eat meat, but there's going to be this side effect. And in this case, if you take a glycine supplement, you may be bypassing that side effect.
Yeah, exactly. Yeah, so basically, like personally, I either consume collagen or gelatin, which is really high in glycine, and doesn't really contain any leucine or methionine. So those are really great. Like they also contain other great amino acids, but they're really high in glycine, which is why it's a great way to actually kind of offset that. So I always tend to try to balance my levels of AMPk and mTOR just kind of from day to day. So when I'm focusing more on stimulating mTOR and kind of focusing on slowing down aging, I'm actually increasing my intake of glycine by either consuming a lot of collagen or glycine along with my other food. And then when I kind of want to build up and want to focus on muscle growth and things like that, like stimulating mTOR, then I'm not really consuming glycine or these sources of complex proteins that I just talked about, because you want to have this balance. Because if you just stimulate AMPk too much, you would just become frail, and you will just not really be healthy, not really enjoy life. So this balance is really important, and it's really interesting how we can use these different tools to kind of balance them and not live in the way that many people do today, because they just consume just all this muscle meat and all these high leucine and methionine products just every single day.
I think it's all about balance and restricting those things that are going to be anti-longevity. And you said glycine. What was the other one you just said? Leucine. Leucine. Oh, right.
Yeah. So I mentioned the leucine and the methionine. Those are the biggest stimulators of mTOR.
Methionine.
No, methionine. So it's methionine. Methionine.
Okay, nice.
Yeah.
Awesome.
So those are really important. But yeah, like I really enjoyed the whole presentation, and yeah, they're just like these concepts of AMPk and mTOR and like slowing down aging and all these things, like they are like so complex, like there's so much to it, and like we could talk for like days about all this, because like there's just so many things to focus on. But I really enjoyed this overview on it. It was really beneficial. And one thing I was also thinking about, like so yeah, you said that keeping cold slows down your metabolism, but the thing is actually that so cold thermogenesis actually speeds up your metabolism, but actually it's kind of the increase in AMPk activity that actually seems to slow down your metabolism and actually cools the body down. So when you're fasting for extended periods, you're kind of increasing AMPk, and then you're feeling colder automatically, and your metabolism slows down. But actually, if you go into a cold shower, you can actually end up shivering, and afterwards you will burn a lot more calories, given the fact that you actually speed up your metabolism. And here it's also interesting because this whole thing about like always slowing down your metabolism is probably the same thing where you want to kind of balance this, because it's really about this whole focus on growth and repair. If you just do one or the other too much, you just will either like become too frail, or you would just grow too much. So like here's also interesting how cold is really good in itself, like cold thermogenesis, and where AMPk stimulation and things like fasting and calorie restriction and just decreased energy intake and all that just seems to kind of slow down your cool down your body, and thus you just feel colder based on that.
Absolutely. And there is quite a bit of evidence behind that. It's all about, how did we evolve back in the day? We didn't have hot showers and tons of food. We were cold, and we were starving. And from an evolutionary standpoint, we evolved these mechanisms to be able to survive for longer. So a lot of it's deep rooted in evolution, everything you're referring to. So thanks for that, Andres, and thanks again, Kamila, for all your thoughts and hard work here. And thank you to the audience for listening. We will be back very soon with another talk. Thank you.